So it's a real pleasure, honor, and very stressful to introduce Pete Schultz. So Peter is the Welch, one of the Welch Awardees this year, and he is the president and CEO of the Scripps Research Institute. He started his undergraduate and graduate work at Caltech with Peter Dervin, working in physical organic chemistry and then selective DNA cleavage, one of the pioneering experiments in chemical biology. He then worked with Chris Walsh at MIT and then began his independent career at UC Berkeley. And I think, I was trying very hard to come up with a phrase for Pete, and the one I came up with during Aranthi's talk was, Pete is never satisfied. And he is just never satisfied with the way biology does something. He really wants to take it to the next level and add a chemical control that is really unprecedented. And the work that I think of in this respect is expansion of the genetic code. And I remember 40 years ago when I was interviewing for graduate school at UC Berkeley, and I met Pete for the first time, and he told me about the idea to add a 21st amino acid to the code. I was honestly just blown away. And when I think about that idea, I can put myself right in the office, right in the chair next to him, where he showed me the idea. And for the students and postdocs, what I wanted to try to convey is that when somebody tells you an idea that is it's as soon as they tell it to you, it seems like an obvious extension, even though you've seen it in textbooks, you learn it in class, but you immediately think that is absolutely crazy and impossible. And then in the next 10 minutes, when he unravels that, oh, there's 3 stop codons, there's suppressor T, all of a sudden, you start to see the impossible become the possible. And that really is something that Pete does over and over again. After I joined his lab, worked on catalytic antibodies, I had this, again, experience where I was looking at the same thing that he looked at, which is how antibodies are made, the combinatorics, and he took that and turned it into combinatorial chemistry, both for organic molecules and materials. So what I think, I hope the students and postdocs see is that in every standard-looking thing, there is chemical opportunity, and I think Pete's ability to identify that, reduce it to practice, and make it impactful is what I always think of about Pete. And this session, for both Welch awardees, we won't do a discussion, so we'll just have the talks, and then after Pete, we'll go to lunch. So please take it away, Pete.
Great. Well, it's a real pleasure to be here and have the opportunity to talk to you, and as a Welch recipient, it's wonderful. I was going to talk about the work we've been doing in our lab for years in synthetic biology, and we have a lot of interesting new results. But then I saw the title of Kayvon's symposium was drug discovery, so I thought I would change the topic of my talk to our translational efforts at Scripps. And it's not a talk that I've given, so this is an experiment. But it's also an experiment for Scripps. Scripps is historically, an institute focused on basic research and technology development, and it has a rich history. I'm not going to go into that, but I think although we're small, we punch well above our weight, and you saw terrific talks from my colleagues, Jeff and Ben. We also have a graduate school that's really terrific. Again, given our size, we're a top 10 ranked graduate school, and we do a lot with educating graduate students in how to do research. But then a few years ago, I was asked to take on the leadership of Scripps, and being president of anything is not something I would wish on anybody. But it is an opportunity to do something new and different and have an impact. And so that's what I looked at. And as I said, Scripps has a tremendous history of great science. And the question is, where next? And is there a way to further expand the impact of the science at Scripps on the public at large? And so Scripps is an ideal place to do that because it's got a rich history of pushing the frontiers and doing things that may be risky, but really move science in a new direction. And so the question is, what would you like to do? And the first thing is you know, fundamental research really underpins all of what we do with chemistry and human health, materials, energy, and everything else. And you know, I would argue it was a lot easier to get funded when I was a young faculty member than now. And it's harder nowadays to get funding to pursue really high-risk, high-reward ideas. So we need to find new ways to support really innovative ideas and new funding sources. And we also want to accelerate the impact of basic research on the public, especially nowadays when many in the public don't really understand the value of basic research. What we really want to do is make clear the value of basic research to the general public, and that is impacting their own health. And finally, we need a way to make nonprofit research more sustainable, okay? So we're not just simply dependent on, you know, philanthropy or the federal government to fund our interesting and exciting ideas. We really want to create a new model for how we begin to fund science. So those were the ideas I had, and that's where we started. And so I'll give you a progress report on where we are. And let me talk about, as a basic research institute, our ability to accelerate the impact of basic research on the development of new medicines. We've heard a lot today about how to do that. A lot of discoveries are made and have been made in the nonprofit sector. In fact, like Stuart said last night, it's the golden age of biology, okay? We're lucky. But surprisingly, the development of new medicines has lagged pretty far behind our understanding of human disease. And why is that? And I would argue one major reason is discoveries are made largely in the nonprofit world in general. We've heard a lot of exceptions today, doesn't have the tools to turn those discoveries into clinical stage drugs. So that happens in pharma, but pharma has very distinct motivations, cultures, and processes. So that creates just these intrinsic barriers. And how do you overcome those barriers? Well, you have an idea, you go and try and raise money from the venture community, and then if you convince them, they have to hire a CEO, CSO, build a lab, and hire the people and reduce that idea to practice. And then if they're lucky, they partner that with pharma and you start all over again. That's also an intrinsically inefficient, expensive, time-consuming process. So we asked, the question was, could we build within Scripps the ability to go all the way from a discovery at the bench to a new medicine, put that medicine into people, and show clinical proof of efficacy in humans? And if you do that, what you've done is you've accelerated the whole process. It's like shopping on Amazon, okay? You go from your computer key to a package at your door. So that's the idea. And if you can do that and go all the way to a clinical stage asset, the value creation is rather large. And the beauty is you can actually reinvest the value you've created with the returns from the partnering revenues into the research enterprise and make the whole model self-sustaining. So you solve two problems at once. You solve the problem of accelerating the development of new medicines and creating a new self-sustaining business model for nonprofit research. So that's what we wanted to do. How did we do it? Well, I had a little bit earlier started a nonprofit translational research institute called Calibr-Skaggs now, and I happen to be president of that. I happen to be president of Scripps, so it was pretty easy to merge the two, okay? And so we created a new division, translational arm of Scripps called Calibr. And, you know, right now it's about 150, 160 people. We have collaborations with major foundations and pharma. The leaders are Gates and Abbey. We've actually advanced to date 14 programs into clinical development. We advanced 4 this year alone, and we have 5 in IND-enabling studies, which is pretty unusual for a nonprofit research institute, okay? We operate, the one rule is we operate completely autonomously. So although we have these collaborations, everything we do is executed by us, okay? The project inception, the execution, and the clinical development. So it's a completely autonomous operation, and we own all the IP. So there's a huge degree of freedom in what we want to do. And to date, we're starting to see the return of not only the new medicines into the clinic, but the business model, because in the last 5 years, we've actually received roughly $500 million through this enterprise, which we've reinvested in moving assets further into the clinic. Ultimately, we'll reinvest that in all of the research enterprises Scripps. So what do we have? Well, we have technology infrastructure. So we have Dell libraries. We have about 160 billion member Dell library. That's a novel 5-cycle library, so it has very large chemical complexity. We have technologies to make tri- and bispecific antibodies. We have the largest repurposing collection in the world, which we built with the Gates Foundation. And we have platforms for long-acting injectables and ADCs and so forth and so on. So we've built a technology infrastructure that allows us to move very quickly once we have an idea. This is the pipeline. I'm not going to go through all of these. One thing I've never been accused of in my career is to be overly focused, okay? And that's reflected here, okay? But the idea is not to do a one-off biotech, sell it, and do it again. The idea is to build a long-term sustainable pipeline so that every year you create new medicines. So let me give you a few examples. And if you're going to do this, in theory, you should do something that pharma's not doing, okay? Because it's tough in the nonprofit world to compete with the resources of big pharma. So if you think about medicines, a lot of medicines slow the progression of disease, almost none stop disease, and none reverse disease, okay? So the question we begin to ask is, can you actually, for the first time, make medicines that actually reverse the disease process and restore normal function? And so the leading cause of death globally is heart disease, okay? I see a cardiologist twice a year, so I always work on things that I have a personal interest in, okay? But, you know, there's 2,200 deaths there's 2,200 myocardial infarctions every day in the United States. So you get a heart attack, part of your heart dies, okay? And then ultimately, although the mortality has been improved, it ultimately leads to heart failure, okay, and death. So we ask the question is, can you actually intervene in that process and repair the damage? And how do you do that? Well, there's not obvious stem cells in the heart, but it was known from other studies that if you activate the YAP pathway, that you can actually put cardiomyocytes into a transient proliferative state and proliferate cardiomyocytes. It was done genetically, which is hard to do clinically. So we just simply looked for novel YAP activators, and we found some. The one that I'll talk about acts through RAD. And we asked whether we could actually turn that into a drug, and we did through a lot of great medicinal chemistry efforts. And we went into rodents and pigs, but the pigs are the best model for human heart disease. You can actually transplant a pig heart into humans. So we went into pigs, and we gave pigs a myocardial infarction with a balloon. And you can see you cause a huge amount of damage. Okay, this is tomography imaging the heart 3 days after infarct. And the yellow is damage, which you can measure by perfusion. And 30 days later, you see almost complete heart failure in the pig. If on day 3, you give 1 intrapericardial injection of the drug, here's day 3, so you see you actually have caused a lot of perfusion damage. But if you give the drug at day 3 and you go back 30 days later, the heart is pretty much returned to normal function in terms of ejection fraction, in terms of cardiac output, in terms of imaging. And you can see that actually a picture's worth 1,000 words. If you give, this is the pig heart, this is the left ventricle. If you damage it by cutting off the blood supply here, you see it beats asynchronously, right? This part of the heart's dead. You give 1 treatment of the drug and you come back and look at heart, normal beating. It's almost completely repaired. So we hope to have this actually in human trials in the next 16 to 18 months, and it really could have a huge transformative impact on heart disease. Can you do the same thing in the lung? Respiratory disease is the 3rd leading cause of death in the world. Okay, COPD, cancer, and so forth and so on. Can you actually repair the lung and reverse the damage in the lung? And in the lung, you actually do have lung stem cells. They're called AEC2 cells, okay? And they differentiate into the gas exchange cells, AEC1 cells. And what we found, we again did an unbiased screen for molecules that would expand AEC2 cells. We found the molecule, and what it does is it inhibits a protease that proteolyzes autocrine factors that are responsible for AEC2 proliferation. So you inhibit the protease, you proliferate AEC2 cells. They turn into gas exchange cells. They do this in every animal model we've looked at, and that's because it's lung repair. Okay, fibrosis, acute lung damage, okay, elastase damage, and so forth. So this molecule right now is just finishing up phase 1 human trials in the Fraunhofer Center in Europe. It's the leading IPF center in Germany, and so that will be, we're moving into a phase 2 trial in IPF patients to try and actually restore the damage to the lung in IPF, which is a horrid disease. The average mean lifetime expectancy is 5 years once you're diagnosed. So we're excited about this. We think this could actually be applicable to a huge number of respiratory diseases. Can you do the same thing with the gut? Crohn's disease and ulcerative colitis are horrible diseases, and what happens is you get a breakdown in the mucosal barrier and your microbiome bleeds into your systemic circulation, and so you get inflammation, and the way we treat that is with anti-inflammatories. That doesn't solve the problem. The problem is the barrier breakdown. So can you actually repair and regenerate the intestinal epithelial barrier? And so there's an obvious way to do that. GLP-2 is an intestinotropic peptide that has anti-apoptotic, anti-inflammatory activities, regenerative activities. It's not a great drug, so we actually used some peptide engineering technology we developed that allowed us to make a very stable, very long-lived GLP-2 that could be used as probably a once every 2-week injectable. And you can see here in a mouse model, if you take the intestinal epithelial and treat the mouse with DSS detergent, you basically destroy it, and one treatment of the drug repairs it, okay? This has now been through a 120-patient phase 1 trial, and what we see is best-in-class PK. We may be able to use this out for a month. In fact, we're developing it in addition to for Crohn's disease for EED with the Gates Foundation. But you see very good safety, and the other thing is you see an upregulation, a very strong upregulation in the biomarker L-citrulline for barrier repair. So we're excited about this. I think we'll start the phase 2 trial next year. We're also working in cancer. We heard a beautiful talk by Wendell yesterday. We're taking a similar approach towards a controllable CAR T cell. So we make a universal CAR T, and then we have an adapter molecule such that you can target this CAR T to any tumor with an adapter. Importantly, you can dose the adapter and control the CAR T activity. And by doing so, we carried out a phase 1 trial in CD19 positive blood tumors. And we showed that we get the efficacy of the conventional CAR Ts, CD19 CAR Ts, but what you do get is significantly enhanced engraftment because you're not getting T-cell exhaustion and you can control activation. And you actually get significant improvements in safety. And based on that, we now can go into solid tumors where the safety issues are far more significant. And so right now we're dosing patients with breast cancer with a novel CAR T antigen. We're starting a clinical trial, a pretty much basket trial in autoimmune disease later this year. And we're turning the whole thing into an in situ platform by creating NEPA and lenti delivery vehicles so you don't have to do cell therapy. Good friend of mine has bladder cancer. Bladder cancer affects roughly 80,000 men, 80,000 people a year. You're treated with BCG. So I would argue BCG was the first IO treatment for cancer. Okay, but it's a horrible treatment. Okay, it's not pleasant at all. You have to have it repeatedly. And roughly 40% of patients are either unresponsive or become unresponsive. So what do you do about that? Well, the response to BCG is really driven by interferon. And so there's a large positive correlation of interferon levels with response to BCG. The best way, probably the best way to upregulate interferon clinically is a STING agonist. So we made a STING agonist that actually is competitive with cyclic GAMPT. And it binds in the active site roughly with a 1 nanomolar affinity, but importantly, in contrast to most other STING agonists, it hits almost all major human STING variants. So we've run this through a series of xenograft studies, both systemically and now in the bladder. And so the idea is you can avoid the systemic toxicity of STING by simply delivering this as an intravesical delivery directly into the bladder. And so we now have a formulation that allows us to do that with upregulated cytokines in the urine, but not in the systemic circulation, that are about on the frequency that you'll need to dose once a month. And so we think this actually, in combination with some of the other recent developments in bladder cancer, could be curative for non-muscle invasive bladder cancer. There's another disease that's an unmet need. It's called myelodysplastic syndrome. And this is a disease where there's a defect in hematopoietic differentiation on the way to blood cells. And so what happens is you get a stop before red blood cells and platelets, and that stop in 35% of the patients ultimately turns into cancer. okay? And so, you know, how do we treat MDS? It's really with cytotoxics, okay? So we said, well, maybe a more benign approach to treat MDS is to actually just look for molecules that overcome that differentiation barrier, because we're working on molecules that control cell fate. Why not induce differentiation? So we used MDSL cells and carried out, again, an unbiased screen and just looked for differentiation in the red blood cell based on markers. And we found the molecule, and we've actually, through a fair amount of effort, turned that molecule into a really potent, or sub-nanomolar potent molecule with excellent PK and very good TIs, therapeutic indexes. So we're marching this now into IND-enabling studies, but it works broadly in MDS in low-risk and high-risk patients. We've actually used it in collaboration with a collaborator at MD Anderson and showed we induce differentiation in 80% of the patient samples we get. So again, we're pretty excited about this. The other aspect of this is, you know, we think about medicines, and we're talking about medicines that impact us, okay? But there's the rest of the world. And, you know, one of the biggest advances in cancer therapy in the last 30 years has been checkpoint inhibitors. But checkpoint inhibitors are very expensive, and you use biopsies before you get them. And so 80 to 90% of the world doesn't have access to the best cancer therapy that's been developed. So what can you do about that? Well, ideally, what you want then, if you want to go into low and middle income countries, is an oral, small molecule checkpoint inhibitor. And because of our relationship with John Martin and Gilead folks, we were able to actually in-license an oral PD-L1 inhibitor that dimerizes PD-L1, once-a-day pill, under really shockingly benign terms, because they were interested in us working with them to develop it. And right now we have that in a phase 2 trial in India. And the idea is it's so cheap and safe that you don't even need a biopsy. If you have non-small cell lung cancer, you just take the drug, and 30 or 40% of the people may benefit, okay? And it's going to be cheap. So we think this could impact, you know, cancer globally and kind of overcome some of the inequities in the world. But also, it may be better tumor penetrant, and it may be better in combination with other small molecule oral drugs for cancer. So it may actually benefit us as well. So we're also, clearly everybody in the world now is thinking about metabolic disease with the GLP-1s. And the fact that you can actually very effectively treat obesity with GLP-1 agonists, and you impact a huge number of other diseases, okay? From liver disease to diabetes to neurodegeneration to osteoarthritis. And now people are developing far better drugs. They're the dual agonists that hit GLP-1 and GIP, and now the triple agonists that also had glucagon receptor agonism. The problem with all these drugs, these dual and triples, is they're all injectables. And I don't know, everybody says injections don't hurt, but I take a PCSK9 injection every 2 weeks. Maybe I don't know how to inject myself, but it's painful, okay? So why not make an oral drug so people like me who want to lose 15 or 20 pounds don't have to fool around with that? So we did. We used, again, our peptide engineering technology to make, you know, a 30 amino acid peptide that is more potent than tirzepatide, okay, in preclinical models. But importantly, has 5 times the oral bioavailability of Rybelsus, Novo's GLP-1. So this is now in clinical trials, okay? And we think it's actually going to be the first really effective oral weight loss drug once a day. We're also working on triples, and I think we can do the same thing with triple agonists. We'll know that soon. The other thing you're starting to realize now is combining activities of biologically active molecules is really improves efficacy, okay? And as you think about this, you know, people are working on bispecific antibodies, trispecific antibodies, not only in cancer, but now in inflammation and metabolic disease and other diseases. And so, you know, I think this is a huge opportunity, and just by way of one example, if you want to deal with MASH, okay, liver fibrosis, you know, there's two ways to do it. If you give somebody a GLP-1, it helps with MASH because you lose weight. It doesn't have a big effect on fibrosis or lipids, but you lose weight, which is really beneficial. If you give FGF21, you get a very significant anti-fibrotic activity and a lipid lowering. So why not give them both? And so what's the best way to do that? Well, we actually used our tri and bispecific platform to figure out how best to put these into a single molecule. And let me tell you, geometry, valency, all of that really matters. So we usually go through and make about 60 or 80 of these constructs to find the right one. But this one, when you give it in MASH models, it actually works better than if you give FGF21 and a GLP-1 independently. And we think it's because you're actually delivering the two drugs to the same tissue at the same time. So we're really excited about this, and this is going to move into the clinic 2027. There's been a lot of effort. I mean, in the old days, I worked on this with Nat on NRF2, because oxidative damage plays a huge role in a huge number of diseases, okay? Everything from neurodegenerative disease to IBD to reperfusion injury. And so people have been really interested in turning on an antioxidant response via ARE and the transcription factor NRF2. The problem is when you do that, people normally do it with molecules that alkylate the cysteines on keep. And so you're using a toxic molecule to turn on a detoxifying mechanism, which at some level doesn't make sense. There is one good example, omaveloxolone. This is a NRF2 activator. It's actually used in Friedreich's ataxia. It's the drug of choice for Friedreich's ataxia, but it again has tox. It's not CNS penetrant. So we actually looked for a novel way to turn this on. And so what we do is we have a molecule that inhibits an enzyme involved in glycolysis. And by doing that, we build up a glycolytic intermediate that actually naturally activates KEAP1 to control oxidative damage in the cell. And now we have a version of that that's incredibly brain penetrant. We've tested it in IBD, and it works as well as anything. It works as well as a steroid in IBD. And now we're looking at it in ischemic reperfusion injury in the heart. And also, we're carrying out Parkinson's preclinical animal models with this molecule. One of the things I really like is we're working on Botox. Botox is interesting because it's used cosmetically, okay? It's got a heavy chain which delivers the toxin, and then it's got a light chain that's an incredibly selective protease, 16 amino acid recognition site. So we and Dave Liu simultaneously asked whether we could just reprogram the specificity of this protease and make very sequence-specific proteases. We took a little different approach to it than Dave. We actually did a stepwise conversion. This protease normally cleaves SNAP25. And so what we did is we converted it to a protease that cleaves synuclein. And there's like 15% identity between the two. So we did it stepwise. Change 4, change 4, change 4, change 4, and then made subsite libraries. And it worked out. We really changed the specificity of this to cleave synuclein. And it does, and it doesn't affect cell proliferation, and it looks to be exquisitely selective. So now the idea is, what else can you do? So I called Kayvon, and he said, we'll go after the RAS tail. We're cleaving the RAS tail now. And then well, why don't we go after MYC? We're cleaving MYC now selectively. But the one I'm really excited about is NaV1.7. So NaV1.7 is in the same neurons that Botox goes to, sensory neurons. So all the delivery, all the clinical, everything's known. But NaV1.7 is the one protein that humans who have mutations in NaV1.7 are insensitive to pain. They are the coal walkers who can walk on hot coals and not feel pain. And so it's the best pain target out there. And so wouldn't it be nice if you could just actually take Botox and instead of, getting rid of wrinkles, if you have back pain, Botox gets rid of it. You have joint pain, Botox gets rid of it. Gout gets rid of it. Burn, get rid of it. You have surgery, they just Botox your site of surgery, no pain, okay? Now we actually have a Botox that's cleaving NaV1.7 better than any protease activity we have, okay? So I think this could revolutionize the treatment of pain and be completely non-addictive and give you the sensitivity you need to know if you're burning your hand on the stove. We also do, just finishing up, we work a lot in diseases of the developing world. The biggest killer out there is TB. A quarter of the world's population is infected, and 1 million people die every year for as long as we know. And there are drugs to treat TB, and there are drug regimens of 3 drugs, but the problem is you got to take them for 6 to 9 months. So what happens if you're taking a drug that's an awful drug and you feel better after 2 or 3 months? You quit taking it. You get drug-resistant TB. It's a huge problem. So we actually, with Cornell, carried out a screen for killing TB inside of macrophages, which wasn't so easy. And we found a molecule that modulates adenyl cyclase, which modulates steroid levels, cholesterol levels in the cell. And what's shocking is it doesn't have much activity on its own, but if you combine it with a standard of care, you sterilize mice completely in 10 weeks. So now this has been fast-tracked. It's finishing up phase 1 study with the Hughes Foundation, and they're accelerating into a phase 2, phase 3 study in the next 2 years, because it could actually change TB from a 6 to 9 month treatment to a 2-month treatment, which will have a huge impact. We're also making long-acting drugs. MMV, Medicine for Malaria Venture, did a survey of physicians in sub-Saharan Africa. Would you rather give people a vaccine, a once-a-day oral pill, or a long-acting injectable? And 95% of them said a long-acting injectable, because you don't have to boost the vaccine, so you don't have to worry whether they're going to come back. You don't have to worry whether they're pill compliant. You give it, protect them for 3 months during malaria season, HIV, you're done. So there's a huge interest now in long-acting drugs. So we looked at this with HIV, and the most impressive drug, I'm surprised Kayvon didn't get somebody to talk about it here, is lenacapavir, which if you give lenacapavir to somebody, you treat their HIV. But importantly, if you give it to them, they cannot contract HIV. So it's a chemical vaccine for a disease that we can't deal with. So the problem with lenacapavir is it needs a partner drug so you don't get resistance. So we actually were lucky enough to make a prodrug of islatravir that in monkeys gives protection for roughly 6 months. So this is now, we've partnered this with Gilead, and they want it to be the partner drug for lenacapavir, and it's just finishing up phase 1 studies, and everything looks really great so far. We've done the same thing for malaria with MMV. The idea is can you actually give somebody an injection at the beginning of the malaria season, have it last for 3 or 4 months, and have them be completely protected from malaria? So we took atovaquone, which is a very potent approved anti-malarial, and it has very low intrinsic clearance, so we actually made a prodrug of that. And now it looks like that, if you give an injection, and it's being developed by MMV, if you give an injection at the start of malaria season, mosquito season, you'll be protected for the whole season. You get an injection once a year and you're done. We think we can do the same thing for Lyme disease. So you get an injection at the beginning of the summer, you're protected for 4 months. Don't worry about a tick bite. That's a work in progress. And finally, you know, what did we learn from COVID? The answer is damn near nothing, okay? Because, you know, H5N1, the reservoir now is huge. It's not only in fowl, it's actually in mammals, and it spreads like crazy, okay? And it's not whether there's going to be a pandemic, it's when is there going to be a pandemic. And there have been roughly 900 cases in the last 20 years. You know what the mortality rate was? Half the people died, young and old alike. So they're predicting in the US, before you get to a vaccine, there are going to be 20 million deaths. Okay, we're not working on this. We had a $100 million grant to work on COVID. The one virus they said we could not work on was flu, H5N1. So the problem here is a bigger problem. You got to worry about COGS because you got to give drugs to a lot of people, and you need a drug combination. So right now, we're kind of on the tail end of making an injectable drug that's really low cost, that's 2 drugs, so you don't get resistance, and you're protected. So if you see somebody, you take the drug, you don't get the flu, or you get treated for the flu very effectively. So we're working really hard on this, and it's shocking that this isn't a major problem that's being dealt with by the larger community. So that's what we're building, and the last slide is you know, the good thing is we're impacting medicine, okay, I think, and we're doing things that pharma doesn't do, certainly in low and middle income countries, okay, you know, and it's a good thing to do. If there is a St. Peter, you got a really good story to tell, okay? But at the same time, as you move drugs to clinical proof of concept, the royalties and milestone payments go up by a factor of 20, which to the institutions is really important because in general, institutes don't benefit from their successes in translational research. But if you actually can internally go to this phase, then all of a sudden you're getting back returns. And you only need a few, you only need one if you're our size, billion-dollar drug where you have an 8 or 10% royalty to all of a sudden make your life easy. And so that really is an important part of the model is now as you succeed, you get more resources so you can take on bigger problems and actually grow your impact in kind of a flywheel-like model that's self-sustaining and grows. And so I think we're well on our way to doing this, and I think if we're successful, it could be a model other folks follow, especially in these times where, you know, federal funding can be a significant concern. And I'll end with the leadership team at Skaggs, who are tremendous people to work with. They're a lot of fun, and they're hugely dedicated to human health. And I really want to point out Mike Bolong, who's an associate professor at Scripps, who I work with closely on all the regenerative medicine work. And he's one of the absolute most gifted people I've ever had the privilege of working with, so I just wanted to point him out. And thanks for your attention.